Hello, today we are going to discuss DNA replication. Some information about life. All life reproduces, all cells divide and multiply, any organelles, other aspects of the cell, and DNA must be replicated when a cell goes through reproduction. Here is 3 different images of DNA. The first 2 images to the left and the middle one, that's how it is easier to see and understand what DNA is, or what it's made of, but the double helix structure, which is the image on the far right, that is what DNA in reality looks like all the time. So, important part about a DNA is that it's made up of nucleotides. So a nucleotide is 1 phosphate, 1 deoxyribose sugar, and then 1 nitrogen base, which is either adenine, thymine, guanine, or cytosine, and they're just abbreviated with the first letter of that nitrogen base. DNA is also referred to as a ladder because there are 2 strands. It's double-stranded, and it looks similar to a ladder. The part that is holding the rungs of that ladder together, those are called hydrogen bonds. You can see them as those dashed red lines in the left image. They're a very weak kind of bond, but they are still a bond nonetheless. Alright, so there are 2 different kinds of nitrogen bases. There are purines and there are pyrimidines. Okay? Adenine and guanine are purines, while cytosine and thymine are pyrimidines. Essentially what you need to know is that adenine and thymine, so A and T, they always bond with one another, while guanine and cytosine, they are always bonding. You can see that purines are slightly larger in structure than pyrimidines, and that's, I kind of think of it as a way to remember as backwards. So if a larger molecule has the shorter name, so adenine and guanine are larger molecules, but they have the shorter name, which is purine. Pyrimidines, of course, are the opposite, so the longer name, smaller molecule. So, DNA replication is made possible by enzymes. So first, what is DNA replication? The process by which a double-stranded DNA molecule is copied to produce 2 identical DNA molecules. Seems pretty self-explanatory, but I thought it was important to include a definition. So replication is an essential process because whenever a cell divides, the 2 new daughter cells must contain the same genetic information for DNA as the parent cell. Now, enzymes, okay, that's what makes DNA replication possible. Enzymes are proteins, so we talked before about how important proteins are. Enzymes are a kind of protein. So enzymes are a substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. So basically, enzymes make reactions where pain is possible. And you can see in the rotating GIF that all the different ways that the enzyme could be affecting reactions. Okay, so here's what DNA replication is going to look like. So again, you have 1 parent strand, and that is going to literally split in half. And then 2 new strands are going to attach to each of the split in half parent strands. So you can see the purple is the original or the parent strand, while the new green or teal that is attached to it, that is the new DNA or the daughter strand. Okay. The picture on the right shows what that would look like in the spiral form, and the bottom left picture is just showing you the same thing but in a different way. Okay, the first step of DNA replication. All right, DNA helicase, so an enzyme, meaning it's a protein, unwinds the double helix of DNA, which breaks the hydrogen bonds, so the middle rungs of those ladder, that thing that holds them together, those hydrogen bonds, those are now breaking up and separating purines and pyrimidines from each other. In the GIF on the left, that is two different DNA helicases going in opposite directions of each other. Other binding proteins make sure that these separated strands don't accidentally reconnect before DNA replication is complete. Okay, the second and third steps of DNA replication. An enzyme known as RNA primase adds a short segment of RNA, which is known as the RNA primer, to each of the separated DNA strands. The RNA primer then acts as an indicator or starting point for DNA polymerase, the third enzyme we're talking about, to begin adding the appropriate new nucleotides that are complementary to the parent strand. So again, looking at the GIF on the left, that RNA primer was just placed, so then the DNA polymerase knows, hey, I need to start adding nucleotides from what was indicated to the enzyme, and we'll see it coming again here, and then it starts matching up the appropriate nucleotides. So if it sees an A or adenine, it's going to match A binding to it. If it sees a cytosine or a C, it's going to match a guanine or G to it. Then, so our DNA has been broken up and separated to allow the DNA polymerase to actually put in those new nucleotides. So the fourth and final step is that we have to seal the two new DNA strands together. And the thing that does that is DNA ligase, which is the last enzyme we're talking about today. So DNA ligase seals the newly formed strand of DNA to be one collective strand and links the new strand to the original or template strand. So original, template, parent strand, they all mean the same thing. You can see again in the GIFs on the left how those essentially match up and reform hydrogen bonds, reconnect phosphate and deoxyribose together, so the side parts of the ladder together. And yeah, that is essentially DNA ligase's function. So here are all the steps of DNA just on one slide, nice and neat. Again, essentially it is one enzyme, DNA helicase, unzips the DNA, RNA primase adds an RNA primer, which tells the DNA polymerase, the thing that actually adds the new nucleotides, where to start adding nucleotides, and then the ligase seals everything back together. And then I have some last images here. So the GIF on the top left is what this process actually looks like molecularly inside your cells. The image on the right shows you what it looks like in a more clear-cut, cartoony way, but that way can be easier to understand as well. The image on the bottom left shows what the difference in DNA replication is between prokaryotic DNA and eukaryotic DNA. So in eukaryotic DNA, it's one big strand, okay, as opposed to prokaryotic DNA, which is a circle. So when these openings of the DNA helicase happen, they're not just happening in one part of the DNA and going from one side all the way to the other. They're happening in multiple different spots, which, as it's labeled in the diagram, replication forks, and that's just so the process of DNA replication can happen a lot faster. And yeah, that's just a unique aspect of eukaryotic DNA and its replication. Thank you for watching. Please share and subscribe, and remember to spread